Good morning students, welcome back to botany sessions, and in these videos we are going to discuss hybridization. It is one of the topic of plant breeding, 12th chapter, hybridization. Next, what are the types of hybridizations? Then heterosis and its types. The next, mutation breeding. Then polyploid breeding. So these are the topic now we are going to discuss. Then first one is hybridization. Then what is called hybridization? Hybridization, it is one of the important tools in the improvement of crop plants. It is a technique or it is a new method. It is a tools for production of new crop by crossing of one or more plants with more desirable characters. These plants are called genetically unrelated plants. If you take genetically unrelated plants, for example, it is a parent one, it is a parent crop, it produces flowers at the tip. It is a parent one. Then crossed with another parent, it produces flowers at the axils. So these genetically two different or unrelated plants are involved. Either maybe same species or maybe different species or different genera or same genera. These one or more genetically unrelated plants are crossed together or brought together to form and first offsprings. These are called hybrids. It is a hybrid plant. These hybrid plants, it possess both the parental characters. These possess both the parental characters are present in the one plant. See, to select a desirable characters of the plants. What are the desirable characters? Increase the yielding, growth rate, increase the protein quality, amino acid quality, to reduce the time of crop maturity, and it shows disease resistant to the insect, pest, drought resistant. These are the desirable characters. All this desirable character, it does not possess into the single plant. It possesses in the different plant, whether maybe same species or different species. But through hybridization technique, we have the ability to bring together all these desirable characters are brought together into the one plant. These are called hybrid. Hybrid. It is a first filial generations or hybrids. These hybrids, it is an important technique. They play an important role in the plant breeding. So these hybridizations are occurs naturally. Naturally, it was first observed by cotton. Cotton method observed in maize plant. So naturally observed. These hybridizations are naturally observed by cotton method in maize plants. Now, what are the steps that are involved in the hybridizations? Steps in hybridizations. First steps, selection of parent crop. Selection of parent. It is our first steps. Second, emasculations. Emasculations. Third, backing. Fourth one is crossing. Then fifth one is harvesting. Harvesting seeds to raising new crop. So these are the steps that are involved in the hybridization. I will explain one by one. What is the selection? To select the male and female plant with the desirable characters. So selection is a better because that possess a desirable characters. Such plants we have to select it. Selection of male and female plants have to be selected. Those plants which are tested to the homozygosity. What do you mean by homozygosity? If, for example, these are the female plant, it is a male plant. It's both are tested for homozygosity. These are the pure homozygosity. I mean, if you are raising this plant for several generations by self-pollination alone. By self-pollination, if raise the plant for several generations, they does not alter its any phenotypic and genotypic characters. Morphological character, phenotypic, genetic character, it does not alter for the after the 5 generations by self-pollination alone. Such crops breedings are called homozygosity without altering any morphological or genetic characters. It's homozygosity. It is also homozygosity. Such the pure homozygosity plants have to be selected for hybridization. These are the selections of parent. These are first steps to select male and female plant with the desirable characters. Second one is emasculation. What do you mean by emasculations? The process of removal of anther in a bud conditions to prevent self-pollinations. See, it is a flower. Anther, male sex organ, stigma, ovary, yes, calyx, corolla, androecium, gynoecium, stigma, center part of the ovary, this is called female part or also called female reproductive part of the plant. It is an androecium, it's a male reproductive part of the flower. If the both the sex organs present the same flower, they are said to be bisexual flower, they are enhancing self-pollinations. Then what is called self-pollinations? Transfer of pollen grain from anther of a flower to the stigma of the same flower. So to avoid self-pollination, to prevent self-pollination, removal of anther. Anther should be removed in a bud condition by the process called anthesis. The anthesis is nothing but maturation of anther is called anthesis. Before maturation of anther, in a bud condition, we have to remove the anther with the help of scissors or forceps. By this process called emasculations, to remove the anther. And next, third steps, backing. Then what is called backing? To protect the stigma of the female flower. Because how to be protected against of certain any undesirable pollen grains may be chances to transfer with the help of certain external agency, either maybe insect or wind may be involved to transfer pollen grain from another flower to the stigma. So to protect, to avoid the transfer of pollen grain from the another flower, how to protect this stigma of the female flower? By covering it with the help of back, by the process called backing. So with the help of back, to protect the stigma, to avoid to contaminations of another pollen grains. So these are called backing process. Next, crossing. Then what is called crossing? Transfer of pollen grain from the male plant. You have to select a male plant. It's a female plant already protected with the help of back. Transfer of pollen grain from the anther of male flower to the stigma of the female flower. By this process called crossings. As a result of crossing, pollinations, as a result of pollination, fertilization takes place. Fertilization takes place within the female flower. As a result of fertilization, seed formations takes place. Now the seeds gets harvested. Yes, seeds gets harvested and they raise into the new generation which are called hybrid. So these are the steps that are involved in the hybridization technique. The next, what are the types that are involved in the hybridization? Types of hybridization. So based on the relationships of plants that are involved in the hybridization, the types may be 4 types. There are 2 types. Sorry, 4 types. One is intra-varietal hybridization. Intra-varietal hybridization. Second, inter-varietal hybridization. Third, interspecific hybridization. Then fourth one is intergeneric hybridization. These are the 4 types of hybridizations. I will explain one by one. What do you mean by intra-varietal hybridization? The word intra, within the varieties. I mean the cross that made between the plants of the same varieties. This is a key concept. Same variety. A cross that made between the 2 plants of the same varieties. These are called intra-varietal hybridization. For example, cabbage crossed with the cabbage. What's the binomial name of cabbage? Brassica oleracea. Brassica oleracea crossed with the Brassica oleracea. As a result, to obtain therefore, it is an hybrids. These are called intra-varietal hybridization. Similarly, rice crossed with the rice plant. For example of rice, IR8 crossed with IR8. Indian rice, it is one of the miracle rice varieties developed by Indian Rice Research Institute in Philippines. These are called IR8 crossed with IR8. These are intra-varietal hybridizations. The next, intervarietal hybridization. What do you mean by inter? Inter means between the variety. The cross that made between the plants of the 2 different varieties but same species. 2 different varieties but same species. These are called intervarietal hybridization. Inter means between the variety, 2 different variety but same species. For example, cabbage crossed with cauliflower. Cabbage crossed with cauliflower. Cabbage binomial name Brassica oleracea varieties. It is also Brassica oleracea variety of botanies. It is a capitata. Both are 2 different variety but same species. Cabbage crossed with cauliflower. These are the same species but different varieties. Its binomial name Brassica oleracea capitata, one varieties, crossed with Brassica oleracea botanis. These are the examples for intervarietal hybridization. The intervarietal hybridization, it is also called intraspecific hybridization. Intraspecific hybridizations. Another one example. For example of this IR8, how they are developed, two varieties. Beta varieties of rice crossed with D. Geo Wu Gen of Taiwan. It's a Taiwan, native of Taiwan varieties. Beta from Indonesia varieties. Indonesia. The two both are rice variety, same species but different varieties. One is Pita rice varieties from Indonesia crossed with Taiwan variety. That's a dwarf. Actually, it is a dwarf rice varieties from Taiwan. It's nothing but D. Geo Wu Gen. D. Geo Wu Gen varieties. By crossing of these two varieties, to obtain the first filial generation, F1 generation is called IR8. It was developed by International Rice Research Institute in Philippines, developed in the year 1960. In 1960, developed by Philippines by International Rice Research Institute. Now these varieties are introduced into India in the year 1960. It is one of the miracle rice varieties. So the IR8 is formed by the crossing between Pita from Indonesia variety. It is an high yielding variety. Another one varieties of dwarf varieties of Taiwan variety, D. Geo Wu Gen varieties. To obtain a semi-dwarf rice variety is called IR8. So these are the example for intervarietal hybridization. It is also called intraspecific hybridization. The next, third types of hybridization, interspecific hybridizations. It is also called intra-generic hybridizations. The word inter, between the specific means species, between the species. It is a cross that made between the plants of two different species but same genus. Two different species but same genus. Same genus. These are called interspecific hybridization. It is also called intrageneric hybridization. The word intra means within or same genus but two different species. For example, Gossypium cotton variety. Gossypium hirsutum. It is crossed with Gossypium arboreum. Both are cotton variety. Cotton. It is nothing but American cotton. American cotton. Gossypium hirsutum, it's a American cotton. Gossypium herbaceum, it's a Indian cotton. Indian cotton. So the both varieties are crossed to form an F1 hybrid. These are called Devraj hybrid varieties. So these Devraj hybrid varieties that possess both the parental characters, the desirable characters are which is bring together into one plant. It is one example, because Gossypium genus, the same genus but different species. What are the species name? Hirsutum and Arboreum, different species. These are called interspecific hybridization. Another one example, rice varieties. Oryza japonica. It is a Japan rice variety crossed with Oryza indica. It is an Indian rice varieties. Oryza genus, same genus, different species. To obtain as a cell, to obtain the F1 hybrid, these are called ADT-37 varieties, rice varieties. Increase the yielding also. ADT-37 is a rice varieties obtained by a crossing between Oryza japonica, Japan varieties, and Oryza indica. These are the another example for interspecific hybridization, or also called intra-generic hybridization. Then fourth one is intergeneric hybridization. Intergeneric hybridization. Intergenerics, two different genera. The cross that made between the plants which belongs to two different genus or genera. Two different genera. So what are the advantages and disadvantages of this type of hybridization technique? The first hybridization, intra-varietal hybridization, that is useful for only in self-pollinated crop. Second one is the inter-varietal hybridization, that is useful for both self-pollinated and cross-pollinated crops. It is useful for the improvement of self-pollinated as well as cross-pollinated crops. The third one is interspecific hybridization. What are the uses of this interspecific hybridization? That is useful for transfer of gene, the gene that responsible for disease resistance, pest resistant, disease, pest, drought resistance, and well adapted. So the such genes are transferred from one plant to the another plant. So this technique, it is useful for the transfer of gene from one plant to another plant. Which technique? Interspecific hybridization techniques. What are the genes that are responsible for disease resistant, pest resistant genes, or also increase the yielding genes also may be transferred. In intergeneric hybridization, it is a disadvantage. A lot of disadvantages also there. What are disadvantages? The F1 hybrid is a sterile. Sterility of F1 hybrid. F1 hybrid sterility, and the time consuming is more. Next, expensive procedure. A lot of procedure also there. So these are the disadvantages of this intergeneric hybridization, because two different genera are crossed. It is impossible, but of course the F1 sterile hybrid is actually sterile varieties. These are disadvantages. For example, Raphonobrassica. Another one is triticale. These are the example for intergeneric. The word inter, in between the genus, intergeneric hybridization. I will explain one by one. How to obtain Raphonobrassica? The Raphonobrassica is obtained by crossing of radish and cabbage. First example, radish crossed with cabbage. We know that's what's the binomial of radish? Raphanus sativum. Raphanus sativum crossed with Brassica oleracea. Brassica oleracea. These are the two varieties. Species also differ, genus also differ. There are two different genus. Raphanus sativum crossed with the Brassica oleracea. For the diploid set of chromosome in Raphanus sativum 2n is equal to 18. Most of the flowering plants, they are diploid in conditions, two sets of chromosome. Brassica also 2n is equal to 18. At the time of gamete formation, half of the number of chromosomes are gets reduced. So here n is equal to 9. It is a egg. Here also n is equal to 9. It is a sperm. The sperm that carry n set of chromosome, I mean haploid set of chromosome. Egg also contain haploid set of chromosome. Both egg and sperms are fused together to form zygote. These are called first filial generation. N plus n, 2n is equal to 18. It is a first filial generation. It is an hybrid variety. This hybrid variety is actually, it is a sterile variety. Malatithanmai. They do not produce yielding. They do not growth that does not take place. It's a sterile variety. But the scientists are thought to plant to double the number of chromosomes. How to double the number of chromosomes? Doubling the number of chromosomes. How? By using colchicine. Colchicine. Colchicine, it is a chemical substance. It is obtained from Colchicum autumnale. Colchicum autumnale. It is a plant obtained from Liliaceae family. It belongs to Liliaceae family. Colchicum autumnale. So it is a source of the chemical substance called colchicine. To obtain the colchicine from this plant, to apply the tip of these plants, tip of the hybrid plant, that induced to double the number of chromosome. As a cell diploid become tetraploid, double. 2n become 4n. Diploid, tetraploid is equal to 36. So that is a fertile varieties. Such fertile varieties called Raphanobrazzica. First name of the genus of one species, Raphanus. Raphano. Here the genus of second species, Brassica. Raphanobrazzica. I mean the radish produce underground part, the aerial part produced cabbage. These are Raphanobrazzica. It is a fertile variety, tetraploid varieties. It is one example of intergenic hybridization. Then another one example of intergenic hybridization, triticale. How to obtain the triticale varieties? By crossing between two species. One is sickle cell wheat varieties. Triticum durum. Triticum durum crossed with sickle cereals. Triticum durum, it is a wheat varieties. Sickle cereal, it is a dry cereals, rye varieties. This diploid set of chromosome 2n is equal to 28 in Triticum durum wheat varieties. Sickle cell 2n is equal to 14. But the critical, it is already exists in diploid condition. 2n is equal to 4x is equal to 28. The 4x, it is a tetraploid condition. They already exist in the tetraploid condition. Because how they are obtained tetraploid? Two diploid plants are crossed to obtain tetraploid. but it is already exists in the diploid condition. 2n is equal to 2x is equal to 14. They already existed in diploid conditions. Now, crossing of these two plants at the time of gamete formation, half of the number of chromosomes are reduced. Here 2n, n is equal to 14. Here n is equal to 7. But what are the conditions also reduced? 2n. n is equal to 2x is equal to 14. Here of n is equal to x is equal to 7. It is a gamete, sperm, it is a egg. Both are fused together to form zygote. N plus n, 2n is equal to 21. 14. 21 to n. Condition n plus n. 2n is equal to 2x plus x, 3x is equal to 21. So these are the triploid condition. Triploid. But actually it is a sterile varieties. Sterile. They would not give the yielding. But they taught to plant to double the number of chromosomes, doubling the number of chromosomes by colchicine treatment. Colchicine, it is obtained from Colchicum autumnal plants. Colchicine to double the number of chromosomes. Here 2n. Here 2n is equal to here 42. 4n is equal to 42. Conditions. 2n is equal to 3x becomes 6x is equal to 42. These are called hexaploid. Hexaploidy typical. It is a fertile varieties. So the first man-made cereal is called typical. But actually it is a sterile variety. F1 condition, actually it is a sterile variety. They are triploid in conditions, sterile, but they thought to double the number of chromosomes by the scientists. As a result to obtain hexaploid condition, it is a fertile varieties. This is another example for intergeneric hybridizations. Next, atrosis. Next heading, atrosis. The word atro means different, cis means conditions. So, G.H. Shell first coined the term atrosis in the year 1912 in State Board book, 1914 in NCERT book. So, G.H. Shell is the first person to coin the term atrosis. Then what is called atrosis? Superiority of the F1 hybrid performance over its parents is called atrosis. Atrosis, it is also called hybrid vigor. Hybrid vigor. What it means vigors? The vigor that refers to increase in the yielding, growth rate, resistance to disease resistance, pest resistance, drought resistance. They can tolerate the drought, they can resistant to the specific disease, and space no one pest can attack. These are the resistance characters. And also adaptability, yeah, very good adaptability. So these are the characters that refers to vigor or potentiality. The F1, when a plant is said to be vigor, it is observed maize varieties. Observed maize. So the 25% of F1 hybrid maize variety, they increase the yielding when compared to that of parents. How to obtain? We selected two parent 1 maize variety cross with parent 2. Both are maize but different varieties. It is B37 variety crossed with MO17 varieties. Yes. It is one variety. It is a maize crossed with another one variety. It is a corn. So these two varieties are crossed to obtain the F1 hybrids. Yes, it is an F1 hybrid. It is a tall plant. They increase the yielding. Large fruit size. Growth rate also increasing. So these are the first filial generation. These are called hybrid vigors. So superiority of the F1 hybrid perform over its parents in the yielding, growth rate, resistance powers, everything they are dominant over the parent. These are called atrocities, are also called a hybrid vigor. How to maintain this hybrid vigor for several generations? Only vegetative propagation. The vegetative propagation are also self-pollinations. It is a best suited method for maintaining these hybrid vigors for several generations, after 10 generations, to maintain this hybrid vigor, because the characters are never lost, can persist over a long period of time. So the magnitude of these hybrid vigors, because of genetic diversity between the two crops. Genetic diversity is nothing but the genes which is present in these two different plants that increase hybrid vigors, also called potentiality. So based on this origin, nature, reproductive ability, adaptability, these atrocities or hybrid vigors further classified into two types. One is U-atrosis. Then another one is pseudo-atrosis. U-atrosis and pseudo-atrosis. The word U means true atrosis. That's called true atrosis. It is occurs naturally. So it is called inherited ones. Inherited is nothing but they occurs naturally during sexual reproduction. The characters are transferred from parents to offsprings. These are inherited ones. This is called U-atrosis. The U-atrosis are two types. One is mutational atrosis, another one is balanced atrosis. The first one is mutational atrosis. What do you mean by mutational adrosis? It is occurs only in the cross-pollinated crop. Due to the presence of adaptability, dominant alleles, superior dominant alleles. Due to the presence of the superior dominant allele, they can eliminate certain harmful or deleterious alleles or deleterious genes, lethal genes, recessive characters controlling gene, certain unfavorable condition genes. Everything gets deleted due to the presence of these dominant alleles. It is better to understand. For example, line 1, line A, it is a crop 1 crossed with line B, another crops, maize. But here the genes, pairing of genes, capital A, capital B, small c, small d, 4 pairs of alleles crossed with small a, small b, capital C, capital D. Here 2 pairs are dominant allele, 2 pairs are recessive. Here 2 pairs are recessive, 2 pairs are dominant alleles. At the F1, during at the time of gamete formation, among the pair only one alleles are get segregated. These are called of dominance. Here A, capital A, here capital B, small c, small d. It is a one gamete. Similarly, here also small a, small b, capital C, capital D. It is another gametes. Among each pair, only one alleles are get segregated into the gamete. So both are crossed together. Capital A, small a, one pair. Capital B, small b, another pair. Next pair, small c, capital C. Capital C, small c. Next another pair, capital D, then small d. So among each pair, one is a dominant allele, other one is a recessive allele. Dominant, recessive alleles. So these are called F1 or Atrosis, are also called hybrid vigor. This hybrid vigor, it is superiority to the both of the parents. How? Due to the presence of these dominant alleles. Due to the presence of the superior adaptably superior dominant allele, they can eliminate the recessive allele character. The recessive allele characters does not express, only dominant characters alone express. What are the recessive alleles characters say? Lethal, sometimes leads to death due to the presence of these recessive alleles. Harmful, deleterious means harmful characters. And also lethality, recessiveness, certain unfavorable characters, everything are gets eliminated due to the presence of these dominant allele. These are called the mutational atrocious. Then another one is balanced atrocious. Balanced heterosis. What do you mean by balanced heterosis? Due to the presence of well-balanced gene combinations. Presence of well-balanced gene combination, they play an important role in the adaptability. They are more adaptable to the natural and normal condition. It is also play an important role in the agricultures. For example, parent 1 crossed with parent 2. Parent 1, capital A, it is a well-balanced. Parent 2, small a. Both are homozygosity. The balanced alleles. Here also both are recessive alleles. Here both are dominant alleles. It is a well-balanced alleles. At the time of gamete formation, here capital A, here small a, both are fused together. Capital A, small a. These are the F4. So due to the presence of dominant alleles is superior to the parent. So these are called balanced heterosis. In balanced heterosis crops, they play an important role. They are well adapted in the according to the natural and normal condition and also useful in the agriculture purpose also. The last topic, pseudo-heterosis. It is also called luxuriance. It's also called luxuriance. How they are called luxuriance? Because it is superiority over the parents only in the vegetative character, not for the reproductive characters. The vegetative parts are enormous growth, excessive growth, but not in the yielding as well as adaptability, not for the growth rate. Only the vegetative parts, root, stem, leaf alone may be luxuriance growth, not in the reproductive part. For example, why do you call it a luxuriance growth? It is also called poorly fertility. It's a sterile, mostly sterile. In case of fertility, it's a poorly fertility. For example, how they cross, it's a gene combination. Small a, capital B, capital C. Cross, it's a parent 1, another parent, capital A, small b, capital C. So it's one pair of alleles located in the same locus, recessive, dominant, recessive alleles are the dominant. If we cross only ones are obtain the gamete, small a, capital B, capital C. Here, capital A, only one, small b, capital C. Both are together. These are the F1. Small a, capital B, small b, capital C, capital C. Take here one pair dominant alleles are present. Here another pair dominant alleles are present. Here both are dominant. So these are called dominant over pseudo alleles. Dominant over pseudo atrosis. Dominant over pseudo atrosis. Due to the presence of these dominant alleles are more, they are superiority to the parent in only for vegetative characters. Vegetative characters, not in yield, not in yieldings. So these are poorly fertile hybrids. These are pseudo alleles. Thank you.